here before you during this eve of Teachers' Day. Let's be informal. Need not maintain pin drop silence. You can chat among yourself, no issues. Let some of my words go into your mind. Okay, I just wanted to share new insights about a new field of treating patients with bacteriophages. Have you heard of bacteriophages? I see one person already started yawning. Okay. So have you heard of bacteriophages? Have you any idea about bacteriophages? Can anybody raise your hands? Want to share anything, whatever you have known about or understood about bacteriophages? Want to share? Okay. I don't want to disturb you. So, I just want to share some insights about phage therapy with respect to tuberculosis. So, I'm not going to talk about tuberculosis. Basically, I come from a center where we do research with tuberculosis. We are into a field of developing newer therapeutics, newer diagnostics for diagnosing the bacteria. Tuberculosis is caused by a bacteria called Mycobacterium tuberculosis. So our own bread and butter is in researching all the areas around this disease. So actually my center basically is one of the center under Indian Council of Medical Research, ICMR. There are 32 research centers under ICMR. So one of the research centers is in Chennai. We do research in tuberculosis in particular. There are other centers which do research in various other diseases like reproductive health, immunohematology, malaria, tribal health. These are the various other research centers under ICMR. So our center is in Chennai. There are two ICMR centers in Chennai. One is NIRT, National Institute for Research on Tuberculosis, and another one in Chennai is National Institute of Epidemiology. So these are the two centers in under ICMR. We work under the headquarters which is in Delhi. This is my customary way how to, whenever I begin a talk, I usually talk about few lines about where I come from, what we do. So I'm not going to go more about what I am doing in particular. We'll directly dive into this topic of phage therapy. So what are these phages? So these bacteriophages are nothing but viruses. They are inert chemical entities when they are outside a living thing. So they specifically infect a bacteria. So that is why they are called as bacteriophages. And the unique thing about this bacteriophage is their specificity. Certain phages attach or infect only certain family of bacteria only. Like Staphylococcus aureus phages only infect Staphylococcus bacteria only. They don't infect Streptococcus, they don't infect E. coli. That specificity is much more governed in these bacteriophages. That is the unique thing about bacteriophages. So they are viral entities which specifically target a family of bacteria. Among that family of bacteria, there are multiple species are there. That cross-borderism, cross-infectivity among the species of same family exists in this bacteria. That means, among Staphylococcus, one of the species is Staphylococcus aureus. There are other species under Staphylococcus. So a bacteriophage which is specifically targeting Staphylococcus will infect, including Staphylococcus aureus as well as other species of Staphylococci also. It will never infect Streptococci or never infect Pseudomonas, never infect Proteus. That specificity is maintained by this bacteriophage. So that unique property is being used to address the antimicrobial resistance nowadays. So what are the advancement around this bacteriophages? That's what I want to talk, lead into this subsequent slides. So bacteriophages are most, and we have enormous of bacteriophages around us. It's most abundant. It's the highest number of living things around us, more than any bacteria, more than any fungi. So you mind that on our body itself, lot of bacteriophages will be sitting on our body. We will not be knowing it. It won't cause any harm. It won't produce any disease also. they infect that particular bacteria only. Otherwise, they are going to be non-living thing. So that is the uniqueness, another unique character of this bacteriophages. So bacteriophages are potentially a very valuable tool for dealing with this antimicrobial resistant bacteria. So nowadays we are researchers in the world, they are trying to address this antimicrobial resistance by the use of these phages. Because there is a rapid spread of antimicrobial resistance, you must be aware of that one. So the next best option, other than the natural sources, other than the herbal products, other than formulations, other than the oil-based formulations, this bacteriophage-based technique of handling this antimicrobial resistance is the emerging field nowadays. It's coming up. So these phage proteins interact very specifically with unique cellular targets. So what is this cycle? What are these phages and what are its applications? So they are being considered as the next alternative to use of antibiotics to treat pathogenic bacterial infections, very specific for one species, as I said earlier. They are self-limiting particles. So whenever they enter into a bacteria, they actually insert the bacterial DNA inside the bacteria. With that bacterial DNA which enters into the bacteria, it actually propagates itself inside the bacterial cell. It is self-limiting, that it can produce multiple copies of the bacteriophages when it enters its interest of bacteria. So when its DNA enters into the bacteria, it multiplies in enormous, numerous hundreds of numbers. It actually takes away the bacterial protein, uses the bacterial machinery to produce multiple copies of bacteriophages inside the bacteria. Then it lies opens the bacteria and comes out, and it infects the fresh another bacteria which is not infected. So again it goes inside and produces large number of bacteriophages. So that's how it is called as self-limiting. And phages are nowadays being totally does not harm any of our normal flora, because they are specific for a particular group of bacteria only. So it doesn't have any interest on other bacteria. Doesn't have any interest because it cannot infect other bacteria. Only selectivity is there. So there is no chance of affecting our normal flora, unlike our antibiotics. Any antibiotic we have to take inside, it is going to affect our gut microflora. No? That is, you must be aware of it. So that kind of issues doesn't arise with the advent of this bacteria. So there are two different forms of bacteriophages. One is lytic bacteriophages, another one is lysogenic bacteriophages. Just a second. So this lytic as well as lysogenic bacteriophages are being used for different contexts. So in the lytic parts, the left side figure shows its life cycle. The bacteria attaches to the cell surface, bacteriophage. The virus actually enters the DNA. The virus particle attaches to the cell wall of the bacteria. It inserts its DNA. The bacterial chromosome is here, but the phage genome separately, apart from the bacterial nucleus, it's separately outside the bacterial nucleus protein envelope. It actually replicates, produces multiple copies of these phages of itself, and then lies open the cell and releases. It doesn't enter the bacterial chromosome at all. So this is the life cycle of a lytic phages. But in the lysogenic parts, there is another group of phages which takes part in the lysogenic cycle also. They are called lysogenic phages, or otherwise it is called separate phages. Wherein the bacteriophage, when it inserts its DNA into that one, it goes directly, assembles into the bacterial chromosome. So then it multiplies, and the bacterial chromosome it actually integrates into the bacterial chromosome, and along with the phage chromosome, it integrates while it divides into two different cells. The phage chromosome also is being propagated, so the daughter cell also receives the phage chromosome also. So this is the lysogenic part. So this is being used for delivering certain gene products into a bacteria. So that's why the lysogenic phages are being used, never for therapeutic use. So when we want to consider bacteriophage for therapeutic, it must be lytic only. So we actually find out When we isolate the parts, we find out whether it is a lytic parts or a lysogenic parts. If it is going to be lytic parts, that can be considered for therapy, for treatment purpose. Otherwise, the lysogenic part doesn't be considered for any phage therapy. So this is the main drawback of the lysogenic part. So there are phages. When we do the propagate them in the bacterial cell, we can find out whether it is a lytic parts or a lysogenic parts. Only the lytic parts will be considered for phage therapy. This you should be aware of it. So when we go for application per se, it can be used to combat antimicrobial resistance, and for cancer treatment also this phage therapy are being considered to cure infections in immunocompromised and transplanted patients. So why in particular to immunocompromised and transplanted patients? In transplanted patients, when you want to transplant an organ into a patient, their immune system is being down-regulated. They are administered with some immunosuppressants. So when you administer some immunosuppressants, there are more chances for opportunistic infections. So in that case, those opportunistic infections are more resistant to currently used antibiotics. In that case, among those patients who are undergoing any transplantation, in that case, the normal conventional antibiotics never being going to be useful for those immunocompromised patients. So in that case, people are now thinking and started using the phages for addressing this opportunistic infection among the transplanted patients or immunocompromised. So there nowadays many people, many countries, they have started using phages for treating the opportunistic infection among the immunocompromised as well as transplanted patients. So there are several clinical trials happened across the world. In India also around 200 patients have been treated with phages, mostly about immunocompromised cystic fibrosis cases, skin topic applications. These are the areas where the phages being tried in several clinical trials nowadays. So there are very famous phage therapeutic centers also. You should be aware of it. So when you want to develop some interest in handling these phages for therapeutic uses, or you want to do some innovations of using this phages, you need to understand where you can find out good placement. So for that only I have taken this slide. So there are very big center, around 100-year-old center in Georgia, Russia. So this is in Tbilisi. There's a phage therapy center. So this phage therapy center is located in Tbilisi, Republic of Georgia, which is in Russia. This company was founded later 2003, but it actually originated way back in 1926 itself. So they are a company where they are interested in medical polymers and biomaterials. They entered into developing phages, use of phages for medical purpose also. So this phage center is dedicated to treat patients with drug-resistant and difficult to treat infections that do not respond to any conventional antibiotics. So it is a 100-year-old center in Russia. They treat several cases of multi-drug resistant bacterial infection by use of phages. They actually personally they have it's called as a personalized medicine. Whenever a person comes with an infection, bacterial infection, they don't have any options of treatment with any of the conventionally available antibiotics. They isolate the bacteria from them. They actually screen the phages which are specific for that particular bacterial infection, and then prepare a cocktail of phages and administer to those patients, and they have saved multiple of patients. So it's for more than 100 years this center is in existence. They are into this phage therapy for several years. So here it's the right place if you want to start your career, but it's in Russia. But in India also there are some centers coming up now. So this is a website, a phage therapy center. Phage therapy center in it is called Ilyava Center for Phage Therapy Center. It has been renamed like that. So in California, in US also, they have founded a center called Center for Innovative Phage Applications and Therapeutics. In short, it is called as IPATH. So nowadays, in 2018, they have started US people also much more interested in handling these phages for treating multi-drug resistant bacterial infections. So the first clinical trial was focused on treating cystic fibrosis patients who had Pseudomonas aeruginosa infection, which was multi-drug resistant. The first clinical trial was started here with Pseudomonas aeruginosa. So this is our website, US San Diego School of Medicine, and they have a Center for Innovative Phage Applications and Therapeutics. In short, it is called as IPATH. So you can browse this IPATH website and find out whether the options are there for continuing your career in future. So there is one more term called phage bioderm. It was commercialized also, so available in the market. Here they have actually prepared a non-toxic, auto-degrading, perforated biopolymer composite consisting of phages and other therapeutic compounds. This was actually called, another term is called to prepare an artificial skin for burn cases and for wounds and burns with special version for osteomyelitis and periodontal diseases. So for this phage bioderm was marketed also. It was approved by the controller also in several countries. This was brought into effect by market by LA Center in Georgia. So it was developed by a phage therapy center, completed the clinical trials and approved in 2000 for regular use. So these are the other companies. Even in India, I told some companies are there. In India, it is called Azger. It's a phage therapy company. In Montreal, it's PhageTech in Biophage. In Sweden, it is phage AB. In US, it's Excel Pharmaceuticals. In USA, another Intralytics, phage bioderm. So these are the other companies where you can find out the phage therapies are being handled in a very large proportion. And there's one private center in India now it has come up. This called Vitalis Phage Group. So this Vitalis Phage founder actually suffered from a very dangerous bacterial infection. It was multi-drug resistant. He was about to die. Actually, he had an opportunity to visit Georgia. He went there and he was saved there by that center. He took a treatment of 2 to 3 months. He stayed there in Georgia. He got fascinated. He got cured. He got saved also. And then he got fascinated by the use of phages. He came to India and started a company called Vitalis. So there, this company, what they do is, if there are any multi-drug patients here, they isolate the bacteria, send it to Georgia from India. They actually develop phages. They prepare a cocktail of phages, treat against that bacterial infection, and import it to India. So that is being administered to the patient, and many patients, more than 200 patients have been saved by that phage company called Vitalis Phage Group. That is a private entity. It's founded by a person, non-medical person, who was saved by this phages. And there is one living example. His name is Thomas. His wife is a nurse, actually. She's a microbiologist as well. This person actually ended up with an Acinetobacter baumannii infection. It was a very multi-drug resistant. None of the antibiotics was able to restrict the bacterial infection. It was a multi-drug resistant Acinetobacter baumannii. He entered into coma also. So they actually got some emergency investigation new drug certification from the FDA of USA for doing phage therapy. And right before the phage therapy was administered, it was thought that he was within hours of dying. Here just his days are counted at that time. He went into coma actually by March 15th. Phage therapy was administered to him for this baumannii infection. By March 15th, in 5 days, he came out of coma. It was a very favorite among the researchers of phages. He regained his kidney function also, which was lost due to heavy antibiotic treatment prior to this phages. He was continuously tried to be saved for more than 6 to 12 months. No response. Then he went into coma. That time only phages were administered to him. Till now, he did not have any side effects at all. It has not been reported yet now. He became healthy now. So he is a living example of a phage therapy. Within 5 days he came out of coma. But actually that phage which was administered to him against this assassinator, but was also less effective after continuous treatment. After he came out of coma, immediately the bacteria inside him got resistant to that phage which was given to him. So phage resistant mutant is also possible there. This is what deeper we need to understand. Phage resistance mutant is also happening with this bacteria. It soon realizes that the phages are coming to kill it, it develops mutations, so that the bacteria prevents the bacteria to adapt to its cell, prevents its entry of its DNA. So that phage resistant to phage is also happened with this patient also. So then they tried with some antibiotics also, minocycline was administered along with the phages, and later he came out of the infection completely. So this was a very good living example for this phage therapy. So these are the several other clinical trials now being conducted throughout the world with phages, chronic arthritis, life-threatening infections, burn wounds, diarrhea, diabetic foot ulcers, venous lung ulcers. There are several clinical trials being now conducted throughout the world with the use of phages. So coming to phages for TB, that we call it as Mycobacter phages. These phages specifically infect only Mycobacter tuberculosis. So this is we call it as Mycobacter phages. So these viruses that infect Mycobacter alone, TB causing bacteria. So till now no resistance have been detected with the Mycobacter phages with the Mycobacteria. Mycobacter tuberculosis, no mutations have been because the growth rate of Mycobacter tuberculosis is terribly low. Slow. It takes 20 hours for the bacteria to double. Other case of other bacteria like E. coli and that takes just 20 minutes for doubling time. The doubling time for any other bacteria is just 20 minutes. But for Mycobacter tuberculosis, the doubling time is 20 hours. So that will be the probable reason why so far we haven't detected any phage resistance from Mycobacter tuberculosis. So no resistance have been reported yet. They are highly diverse and lytic Mycobacter mainly B29 and B4. These are names of specific phages which can infect MTB are now available throughout the world and being researched for treating tuberculosis, which are more resistant to directly available anti-TB drugs. So how will phage therapy can be employed for tuberculosis? That's what I am describing here. Should be this Mycobacter phage should be able to enter inside the background because TB is an infection where intracellularization is very much possible because the bacteria can survive. The bacteria which is responsible for causing tuberculosis can survive and live hale and healthy inside a human macrophage. So intracellular infection is possible here. So whatever drugs we administer should have the capacity to penetrate, get inside the macrophage to eradicate the bacteria. So some of the anti-TB drugs can do this one, some of the anti-TB drugs cannot do this one. So only those drugs which can penetrate inside the macrophage only can lead into complete cure of TB. So that's why we actually treat TB patients with multi-drug therapy. We use rifampicin, isoniazid, ethambutol, pyrazinamide, a combination of 4 anti-TB drugs are being used for treating TB. Some of these drugs can enter this macrophage. So that is an unique aspect of TB. That's why some people come as a relapse or even after complete intake of drugs. So here, the phages, when you consider this to be used for treating tuberculosis, should have the capacity to enter into the macrophage, reaching the bacteria which is hiding inside the macrophage. So that should be the nature of the phage when we want to consider the use of phages for treating TB. So this particular phage named B29 has the capacity to enter into the macrophage. So we have an ideal combination of phages. Some phages which can infect Mycobacterium tuberculosis can reach inside the macrophage also to get the bacteria and get rid of the cell. So this B29 can getting to macrophage. And so the phage cocktail is actually required actually. So we cannot consider this was a very old slide where we thought that phage cocktail is not needed to target tuberculosis, but we cannot take chances. Definitely we need to have a cocktail of phages for treating TB also. So, and there are reports that mycobacterium surpasses cellular immunity also. So there, the reports are that this was confirmed by several in vivo experiments. These phages which can infect TB, particularly, they don't elicit any cellular immunity. So there are engineered bacteriophages also for treating other mycobacterial species. There are several mycobacterial species. One among them, which is called as mycobacterium tuberculosis, causes TB in human. There are other opportunistic mycobacterial species infection also, like M. abscessus. So they cause a disseminated form of infection among humans, mostly among immunosuppressed. So here in this case, they have genetically engineered these phages to treat the infection. That results are also much more talked about in recent times. And there is one patient called around 15-year-old patient. He was seen with mycobacterium, disseminated mycobacterium abscess infection. This was completely recovered by the use of 3 phage cocktail. So here he underwent bilateral lung transplantation so that his immune system was depressed by some steroids. So he got this infection of mycobacterium abscesses. So the phages were administered intravenous and it was tolerated also. But he soon developed neutralizing antibodies against these phages. So that neutralizing antibody also arises when you administer the phages through intravenous route. So you need to select a different route. When it is introduced into the human system through intravenous, there are chances for development of neutralizing antibodies also. So to circumvent this problem, we can choose another route also by aerosolization or by nasal spray. We can choose another route to reach the infection site. That is also possible. We need to pick up the alternate routes to just avoid the development of neutralizing antibodies. And there is a very famous publication where they have compiled around 20 patients case study, different multi-drug-resistant mycobacterial infections. They treated these phages, these infections with phages, and they found out that around 11 patients got survived, 9 patients died due to other comorbid conditions. They were all at the terminal stage. On compassion ground basis, the phages were given to these 20 patients. 11 patients got survived. All the bacterial conversion had happened due to the administration of the phages, and no phage resistance was observed in all these patients. Neutralizing antibodies were identified, that's what I said, in the serum of the initiation of the phage delivery. So in 8 of the patients, not in all patients. So neutralizing for antibodies problem also arises through administration of the phages when you parentally administer the phages. So what are the results? So to overcome this one, there are other things you all can contribute, like encapsulating with the different polymers, encapsulating the phages with liposomes, lipid encapsulation. This will actually prevent the exposure of the phages in our bloodstream, so that neutralizing antibody development can be restricted. So this kind of things you all can contribute and develop innovations where you can hide the bacteria from our immune system so that the phages can be safely introduced into those infected patients. So some people consider use of immunosuppressants also, but it is unethical actually. Ethics won't allow us to use immunosuppressive just to treat them with phages. So that is another legal chance only. So encapsulation with the different polymers is the best choice, just in case if you expect any neutralizing antibody development. So another thing is you need to prepare a case for administering phage on compassion ground after getting the ethical clearance. So these are the flow by which you can start thinking of using phages for treating the patients. Thank you. Please say that important information I will share with you. Got some insights about phages, guys? I want to ask some questions, I want to clarify something, you want to learn anything more which is not clear to you, you can ask. Does anyone try? Anyone want to try asking some questions related to this phages?
Any questions? There is any immunogenic issues with phage therapy? Immunogenic issues with
Yes, that's right. The presence of neutralizing antibody arises when you introduce the phages in the bloodstream. Around 20 patients, among 20 patients, 8 patients develop this neutralizing antibodies. That is there. That's why when it's in the case of respiratory infection, the other choice is to change the route of administration. Again, they have switched the route of administration to nasal entry, by which they circumvented the problem of neutralizing antibodies. So immunogenicity can be handled by this route, or you can hide it with the
one of the limitations. One of the limitations.
Main limitation is to handle the ethics. Ethics. You need to justify the ethics for the use of these phages. You need to have a lot of clinical trials for developing evidences for the government of India to accept it. So still a lot of research has to be done. Probably mostly in TB, a lot of clinical trial has to be conducted. So now actually what we have is around 50 patients across the country are being treated with phages for TB. This is a new development that has been occurred. For other infections, multiple hospitals have developed phages, phage therapy, updates for other infections.
Doing preclinical studies, is there any specific regulations?
For preclinical, we don't have any specific regulations. Clinical only we have regulations. Regulations are usual. We need to have either IND certification for Indian government. We need to have an IND certification or an import license. Import license which should come from a phage group who are already using the phages for therapy across the country, away from the country. So you should import them phages from them, or you should have an IND certification. For IND certification, you should have a GMP certified lab for testing the purity of the phages. Phages, yeah, actually cost per se for producing manufacturing phages are very lesser. The purification part only takes some amount of cost to purify the phages, because the only way of multiplying the phages, propagating the phages, manufacturing the phages is by the use of living cells only. So of course it will carry the other proteins also. So you need to have a proper procedure to purify them. So the purification cost might escalate the phage cost. But definitely it will be lesser than the antibiotics.
But by 2030,
By 2030 we have to do this
as the government says that end of the TV.
Yeah. But end of this is wrongly understood that eradication. It is not eradication, elimination of TB. There are multiple indicators they have we need to bring the incidence to a particular level. So elimination, they have defined it as need to bring down the incidence from whatever is now it's around 300 per lakh. We need to bring it below 50. We need to bring the mortality to 0%. Catastrophic cost exposure should be very less. So these are the three main indicators by which we can call that as eliminator. It's not eradication. Elimination is also very far in a country like this population. It's very far. It's very difficult. That's all. Thank you, boss. Thank you.
Thank you, sir. Thank you for sharing your research experience with all of us. Thank you, sir. I hold the audience to stay here. We are going to start the session within few minutes. I request the students to stay back and please keep your mobiles in aeroplane mode or in a silent mode. Okay, we are going to start the session within few minutes.